Welcome to the online site visit of the Austrian Center of Industrial Biotechnology. We will guide you through the main topics of our Comet Center acib, which are distributed to several locations all over Austria. What is acib's research program all about and how do we implement it? acib addresses today's global biocrisis and provides solutions to environmental resource and health issues at the interface of academia and industry. A mission-driven focus opens up opportunities for industrial partners and intends to meet societal needs. One of the lighthouse topics of the past few years was the utilization of carbon dioxide for producing new high-value compounds. Our research groups at WuKu Vienna give us some insight.
In the Lighthouse project, we are using a synthetic autotrophic Pichia pasteurus yeast strain that is able to grow on CO2 as a sole carbon source and uses methanol to harvest its energy. This was done by implementing a CBV cycle, a Calvin cycle, to the genome of this yeast strain. But we didn't stop here, we stepped a bit further in the project and we converted this autotrophic synthetic yeast strain into a producer strain that is able to produce organic acids such as itaconic acid or lactic acid only from CO2. And in the next years, we want to even improve the efficiency of these pathways by implementing synthetic metabolisms or pathways to the genome of this yeast strain that can utilize CO2 and methanol as a whole C1 carbon source. In this project, we also collaborated with famous artists from bioart fields and presented our works in an exhibition called the Fermenting Features. The opening was in Vienna last year and it brought a lot of attention from the public. And now actually the exhibition is on tour in Europe. Let's hand over to my colleagues in Graz who use the Bacterium Cypriviadis necator for carbon capture.
To grow our cells on carbon dioxide, we needed to overcome some challenges. As oxygen and hydrogen can explode in a range between 4 to 96%, we need some special devices as well as equipment to ensure safety for ourselves and the laboratory. Therefore, we mix our gases individually depending on the needs of the organism. Through a check valve, we ensure that no gas can go back to the gas bottles. On the other hand, we have a frit to ensure that our cells get as most of the gas dissolved to grow and develop themselves in a better way.
Gas fermentation for CO2 capture is a concept that has led to two startups in the acib environment, Akion and Econutri. Both use the concept to produce peptides that are used in cosmetics, food or animal feed. This noticeably relieves the burden on agriculture and offers an alternative for the increasing demand, especially in the food market. Verena and Helmut Schwab, can we please have a quick look at your premises?
About 2 years ago, we founded our startup company Econutri, and right away we began to produce this reactor system with about 300 liters for pilot studies in fermentation. And the goal is to produce protein products for feed compositions, and later on we also want to make the product available for human consumption.
So the main intentions for the research work in the next period will be, on the one side, to further optimize the process, especially in view also of the downstream processing side, where we will focus on developing the process for producing protein which can be directly used for human consumption. We will also do research in strain improvement in order to have more effective strains, more productive strains, to have a highly efficient process in the final end to be run in the large scale.
The production of food proteins brings me to our area renewable resources. The growing world population leads to an increased demand of resources, and the public awareness of resource-efficient products leads to a paradigm change towards alternative diets, personal care products, and more sustainability in agriculture and production processes. Cultured meat secures the food supply for a growing population and promises a smaller environmental footprint than conventional agriculture. Another research area dealing with sustainable agriculture and biodiversity is also based in Graz. This group specializes in microbiome-based solutions for plant protection and seed treatment.
Microbiomes are in control of all biogeochemical cycles on Earth, and their diversity is imperative for ecosystem functioning. For plants, we know that microbiomes facilitate nutrient acquisition, protect against pathogens, and stimulate hormone production. Thereby, microbiomes offer many opportunities for sustainable and climate-friendly agriculture.
You might have noticed it. Sustainability is a very important aspect for all of acib's research topics. There's even a whole research area dedicated to sustainable production.
We see biorefineries as a really enabling technology for a sustainable bioeconomy in the future. If you think 20 years ahead, we will probably want to use most of our products of daily life produced with our biorefineries from renewable materials. One of the examples we are working on is the production of peptides, which are useful components for medical applications and also for food and feed applications.
acib has successfully engaged in microbial protein production for many years and recently started also activities in the emerging field of animal-free meat and feed production. So far, the main challenges to be overcome were always the bottlenecks in the cellular production machineries. So in the previous funding period, our approaches focused on using synthetic biology tools in order to overcome these bottlenecks. While we have so far focused on enhancing productivities, it is now time to make a leap forward and increase the sustainability of microbial protein production processes. In order to do so, in the upcoming funding period, we will follow up several parallel approaches. This will focus on the one hand to reduce or totally avoid the use of environmentally harmful or toxic substances, such as antibiotics, which are currently used. On the other hand, we will aim to enhance the use of the cellular resources and innovative approaches such as including hybrid modeling and digital twins.
acib is also very active in the field of pharmaceutical biotechnology. A lot of industrial partners such as Boehringer Ingelheim, Sandoz or 3M are long-lasting partners that rely on acib's broad expertise of biopharmaceutical processing and the development of novel therapeutics and diagnostics.
The manufacturing process for most new drugs needs to be developed from scratch. Process development is a time-consuming task which limits the availability of new treatment options. Platform processes, like those used for monoclonal antibodies, can accelerate process development and increase manufacturability of biopharmaceuticals. acib, together with Boehringer Ingelheim, have developed a new platform process that is usable for all proteins, not just monoclonal antibodies, called Cas1. The Cas1 platform process significantly reduces the time it takes to develop a new biopharmaceutical, which increases the availability to those in need. But not only does it shorten development time, it also significantly reduces production costs, which increases the affordability of these new treatment options for everyone. But our journey doesn't end here. Working hand in hand with Boehringer Ingelheim once again, our next ambitious endeavor is the development of a biological production process for peptides. This groundbreaking approach will replace the current synthesis process that relies on toxic chemicals. By incorporating non-canonical amino acids, we unlock new chemistries that enable the treatment of previously untreatable diseases. Building on our current success in continuous manufacturing of bio-nanoparticles, we will develop new processes that will reduce the cost of these new modalities. My colleague Patricia will show you more.
Nowadays, the development of new therapeutics relies on the use of complex biopharmaceuticals. And these complex biopharmaceuticals can be, for example, bio-nanoparticles, which include virus, viral vectors, virus-like particles or exosomes, or even cells can be used nowadays for these, like stem cells. At acib, we have not only the infrastructure, but also the know-how needed for the production of these complex biopharmaceuticals, including downstream, upstream, and analytics. In addition to that, with the available infrastructure and technologies that we have at acib, we can develop novel diagnostic tools. We are now at the end of our short overview about acib's research topics. We hope that you have gained a good impression of our activities, and we will be happy to continue our efforts and contribute to a more sustainable future. Thank you for watching.